Hello, everyone, and welcome back for Chapter 6, Acellular Pathogens. I like to cover this chapter before completing microbial genetics section on gene transfer because in order to understand gene transfer, you really need a background on a lot of the terminology and concepts that are discussed when we go over viruses and their related organisms. As you're about to learn, acellular organisms, they lack many features of a cell, especially the plasma membrane and ribosomes. Without a plasma membrane, you're not a cell, and without ribosomes, you can't express your own genetic information, make proteins, and therefore run your own metabolism. And so, for these reasons, they're considered acellular. The way they function is much like you would think of a hostile takeover of a company. They basically infect a cell and divert the cellular machinery and resources into the production of more acellular pathogens. They include a whole host, pun intended, of different entities, including viruses, viroids, which infect plants, and they're typically much smaller than viruses. Then virusoids, and these virusoids, they're just a single circular strand of RNA. And viruses, as we described them, require cells in order to replicate. But virusoids actually require viruses in order to multiply themselves. So they are even more reliant on other entities in order to be replicated. Then we have the prions. And prions are in a whole different category than viruses, viroids, and virusoids. They are actually infectious proteins. And these proteins can be replicated or reproduced in new hosts, which makes them um, able to multiply. In humans, the hepatitis D virus is an example of a virusoid. It's capable of causing pathology only when hepatitis B virus is present. So this is a basic image of a virus. Viruses are infectious agents. We don't call them cells. We don't call them living organisms. They're composed of nucleic acids, as you can see in the center here. And this, interestingly enough, their genome can be either DNA or RNA, and it's going to be enclosed in a capsid, a protein sheath that protects that nucleic acid. Viruses are incredibly small. Now, we said that the average size of a bacterium is somewhere around zero point 0.8 microns, micrometers, to about 2 micrometers. You can get larger ones, but that's, about the average size. The slightly smaller ones too. But 1,000 nanometers is equal to 1 micrometer. And so viruses range from incredibly small sizes all the way down to 20 nanometers, which is 0.02 microns, so 1/100th the size of a bacterium, to about the size of one micrometer. Again, they are acellular. You notice we don't see a cellular membrane here. There were no ribosomes present. So they're not really fully alive. They're basically just genetic code that is stored in a medium for transfer to a different organism. Here we can see some of the common sizes of viruses and how they compare to other organisms. From very small viruses, the bacteriophages F2 and MS2 are only about 24 nanometers. And then we can get into much larger viruses, of course. The poliovirus, 30 nanometers, rhinovirus, 30 nanometers, adenoviruses, 90 nanometers, rabies virus, about 170 nanometers. Here's a prion right here, about 200 nanometers in length. And a lot of these different viruses that I listed so far can, except for the bacteriophages, can infect animal cells. The bacteriophages, T4 bacteriophage, this is a specific type of virus that infects bacteria that we'll get into more detail about its structure. You can tell it's a lot more different than the prior ones we just looked at. and that's about 225 nanometers. The tobacco mosaic virus, which infects tobacco plants, um, it is about 1/4 of a micrometer. And then here's a viroid and a vaccinia virus, so on and so forth. But yeah, we'll dive more into detail later on. You don't have to memorize the different sizes here from this list. You just need to know the general sizes that we have of these organisms, all the way from the very smallest, about 20 nanometers, to some of these very large ones. Here's an Ebola virus that is a whopping 970 nanometers, almost essentially 1 micrometer. And they can get actually quite big. There's very rare examples where they can be quite big. So here's what we call a Pandora virus. These are a new set of viruses that have been discovered that are just enormous. This one can be about the width of an E. coli bacterium, so about 1 micrometers in width, which is absolutely massive for a virus. So, why aren't viruses really alive? Well, like I was saying, they can't reproduce or metabolize independently. They require a cell in order to do that function for them. So, they infect something that's living, and then all of the components that make that thing alive or what the viruses use in order to replicate parts. So, you wouldn't say, if you were looking at our own cells, the cell is the smallest unit that is living in our body. And if you were to break it down, you wouldn't say the plasma membrane alone is alive. You wouldn't say that the Golgi apparatus is alive by itself, or even the nucleus. It's the combination of everything together that makes our cells alive. And nothing by itself is going to meet all of the characteristics of being alive. And the same is true for these viruses. So, if viruses are not infecting a cell, they're basically inert and not capable of, again, reproducing or metabolizing. Just like if you extracted the nucleus from a cell, the nucleus wouldn't be able to do much outside of the cell. And so, yeah. Here we can see different examples of viral structures. We have fully developed infectious viral particles that we call virions. And so when they're fully assembled here, you can see this is an HIV virus, this virion, this is a hepatitis B virion, the Ebola virion, adenovirus virion, here's influenza, and a bacteriophage virion. Virions can have all of the following. All of them have nucleic acids, of course, and a capsid. Some of them can include an envelope. So this is a membrane that can surround the virion. And some of them actually have enzymes like that Pandora. If we go back here to this one right here, that Pandora virus is an example where they have their own enzymes present inside that can do some enzymatic activity, obviously not the full suite of metabolic activities required to reproduce. The viral genomes are incredibly diverse. Everything that we've talked about so far in the three domains of life, archaea, bacteria, and eukarya, all store their genomes as DNA, deoxyribonucleic acids. But the genomes of viruses can be much more varied. They can be either DNA or RNA. They can be single-stranded or double-stranded. So the DNA that are in some viruses are just single-stranded DNA, and they can be linear or circular. So here we have an example of a capsid. And if we're looking all the way in the middle here, it's these lightly colored balls that surround this string of purple color here. That's supposed to be the nucleic acid. And so we have this capsid sheath here that protects that genome. And it's composed of several smaller units that are called capsomeres. Capsomeres build up to make the entire capsid that protects the material. And of course, they can vary in structure and depending on what sort of virus we have here. So in this structure, we can see that it's this very long loop that encases the nucleic acid. And if we look at the outer layer here we have an envelope, this yellowish orange layer here. These are made up of lipids, and we see these green things here. They could be either proteins or carbohydrates that stick out from that virus, and that can help it determine what its host range is. So, what organisms that virus should infect. It can be associated with identification of what cells it needs to infect, or attachment to those particular cells. The host range again describes the species and or cell types that a virus can infect. So here in this box here, we see there's H115 and N19 viruses, and those can infect both humans and pigs. Here we have pigs here with H1N1. You may have heard of that, was what caused swine flu quite a while ago, and H1N2, H3N2, H4N6, so on and so forth. And these are terms that we use to signify those polysaccharides and proteins that are embedded in that envelope that determine what hosts that organism can infect. This is a really important phenomenon for viruses. If you imagine if this H115 can only infect these different hosts here, we don't want that virus particle wasting its efforts if it lands on, say, a cat to try and infect that cat cell, because it would be a lost cause. There are different morphological features for viruses, just like with any other cell, with any cells. One of which is called the helical viruses, and the Ebola virus is probably the typical example of a helical virus. They are long rods. They tend to have a cylindrical capsule. Here's the capsid. You can see that it's going to follow along that nucleic acid for its whole length. And the capsomeres are arranged in a helical pattern. So you can see they kind of go in this helix around here. And the nucleic acid is attached inside. Other examples another example is rabies virus. Rabies viruses are also helical viruses. Okay, here is the polyhedral viruses. These polyhedral viruses are made up of a bunch of triangular faces, and the geometry of such means that you end up with 20 triangles around the entire organism, and they're all equilateral triangles, so they all have the same angle for each of the three angles within the triangle. Examples of polyhedral viruses include adenoviruses and polioviruses. Both can infect monkeys and apes. So this is an important one to be aware of. And now we have the enveloped viruses. Enveloped viruses are ones that are covered with a spherical envelope around the top. So we can see in the center, again, we have our nucleic acid, and then these capsomeres that build up the capsid around the nucleic acid. Here's our envelope and our different spike proteins that are jutting out from that envelope. And this is another important morphology because the flu virus and herpes viruses are enveloped viruses. Moving along, there are viruses whose host range is specific to bacteria. They are called bacteriophages. Phage is another word for virus. So these bacteriophages have evolved to just infect bacteria, and they have a completely different morphology. They look something like maybe a lunar lander or something like that. And if we look at this particular one here, you can see just how complex they are. There are several different specific components that work together in order to make this all work out. So at the very top, we have our capsid, and we have our DNA that's held within this capsid. But all of the other components here, this is all about landing onto that bacterium and attaching itself to the bacterium and infecting it. So we have these tail fibers here, which help it land. We have these pins that help attach itself to, or help, yes, firmly attach itself to the bacterium. The base plate, which supports those components here. And then the sheath is like the tube that allows the DNA to go through. And the DNA actually gets literally injected through this tube and right into the bacterium. So all viral replication is going to follow these schemes here. They can vary depending on what virus there is, on how they do each one of these, but these are the different steps that you should memorize here. The first is that they need to attach themselves. They typically interact with some sort of receptor on the host cell that virus identifies, and that helps make sure that it infects the correct cell and not the wrong cell. And then we have penetration. So one way or another, the virus is going to penetrate the host cell wall under membrane. And this might be the whole virus that gets in. It might just be part of the virus that gets in. It might only be the nucleic acid, but it has to get inside of the cell in order to replicate. Once it's in there, it needs to hijack the host cell and rewire that host cell to start dedicating its cellular resources to making more virion parts. So this is called the biosynthesis step. And finally, we have the maturation step, where all of those virion parts are assembled together to create full viruses. And that brings us to the final step, which is release. Those new viruses are released from the host cell in order to infect other viruses. And this release step can also vary quite a bit depending on the type of virus we're looking at. But those are the 5 steps: attachment, penetration, biosynthesis, maturation, and release. Okay, so again, replication schemes are going to vary depending on the virus, particularly the nucleic acid type and host range. As we discussed, there are single-stranded, double-stranded DNA and RNA viruses, and there's also something called the sense of those strands of DNA or RNA, which we'll discuss later on, which can affect the way that that virus's nucleic acids get replicated, get expressed, so on and so forth. We're going to consider bacteriophages, animal viruses, which include DNA viruses and RNA viruses, and a special group called retroviruses. There are, of course, viruses that infect plants and protozoa, but discussion of these is beyond the scope of this course, so we're not going to get into those. There's an estimated 10 to the 31 different bacteriophages on Earth, which is actually more than there are living organisms, including all of the bacteria combined. If you ever go swimming in the ocean, you're swimming in water that is teeming with viruses that far outnumber all of the other microbes that are present there. So, bacteriophages in particular, their hosts are bacteria. And as we discussed, they have an incredibly complex morphology. They are non-enveloped. We went through the different parts of the bacteriophage on the right here, and we did not cover that there was any envelopes. There are these protein capsids that cover the nucleic acids, but there's no envelope over the top there. There's no envelope that's necessary. Again, the envelope is there, provides some protection, but importantly, the polysaccharides and the proteins that jut out from the envelope allow it to identify its host, and that's not the part that's used for attachment. Again, the tail fibers and the base plate is used. Those spikes there are used. It has a double-stranded DNA genome. So as I said, there's a lot of different types of viruses based upon their nucleic acid structure, but for bacteriophages, it's just a double-stranded DNA genome. And there's two replication mechanisms that we need to cover. Again, that's an important step in the virus, quote unquote, life cycle. One is called the lytic cycle, and the other one is called the lysogenic cycle. Okay, we're going to cover both of those different cycles as they apply to bacteriophages. So the lytic cycle comes from the word lyse, which means to break apart. We've learned about plasmolysis when we learned about osmosis, and that is when the plasmid membrane breaks apart or lyses apart. So what happens here is that bacteriophage lands on its host bacterium, and it's going to inject its nucleic acid, shown here with that green line, into the cell. So we have attachment and penetration. Okay. The tail fibers attach to receptors on the bacterial cell wall. And for penetration, there's a viral enzyme called lysozyme, and it's going to break down part of the cell wall right where it's going to inject its DNA. Okay. And so the viral tail contracts and injects that DNA into the cell, the host cell. Now we have biosynthesis, where the virus shuts down transcription and translation for the host cell and redirects that host to making enzymes, nucleotides, ribosomes, and amino acids for viral proteins. And maturation is going to occur as well, where all of those viral components will assemble, and they kind of auto assemble with one another, much like we discussed with tubulin. The process of tubulin, how it just automatically assembles a microtubule. Or we also discussed with plasma membranes, those phospholipids just automatically line up such that the phosphate groups face outward and towards the aqueous solution, and inward towards the cytosol, and the lipids tails face inward towards each other, huddling away from those aqueous solutions. So they just spontaneously are made such that they just assemble once they're in each other's presence. Now we have the lysis process, hence where this process gets its name from. So once it's used up the cell, it just breaks apart the plasma membrane. It lyses that organism, and it typically kills the host and releases all of those new virion particles, those fully formed viruses. So not all viruses cause death of the host cell. Bacteriophages can go through an alternative cycle here. This is called the lysogenic cycle. And these are called temperate phages, and for good reason. They basically don't go straight to this process of biosynthesis and maturation and release via lysis. Instead, what they do is their nucleic acids get integrated into the host chromosome or host genome. So here we have the circular genome of the bacterium represented in purple, and that viral genome actually gets copied into the host genome. So it integrates. That's an extra step here. We call this a prophage. Remember, pro means before. So just like with the prokaryote, these were evolved before karyotes were created or nucleuses were created. Prophages are before we're going to have a mature virus built. So these therefore are integrated in, because they are integrated into the host chromosome, they're therefore going to replicate with the bacterium, and no major harm is going to occur to that bacterium yet. But just like if you live in a temperate climate, during certain periods of the time, the seasons are going to be different from one another. And when there's the right season, some sort of environmental trigger, this could be UV light exposure, certain chemicals, or other things, it's going to cause induction to occur. And this is what this means is it's going to induce or bring about the release of that viral genetic code, and it's going to result in the continuation of biosynthesis and maturation of the virus particles, and then finally release. So this extra loop, this extra process here is called the lysogenic cycle. So not all viruses perform the lysogenic cycle. Not all bacteriophages do this. Some of them just have a lytic cycle, but some of them have this other cycle where they can wait for a better time in order to be induced. And this does provide some evolutionary advantages to the viruses that do this. If you imagine, instead of just making enough viruses that one bacterium can support, and now we have two, or however many it goes on for until finally induction occurs. There are a couple of major consequences of the lysogenic cycle on bacteria. The first one is the bacterium may acquire immunity to reinfection. So if we see in this example here, here's a bacteria. It got infected with a bacteriophage that conducted a lysogenic cycle. It integrated its DNA into the host genome, and that bacteria along the way may have picked up some immunity to reinfection. And so it survived this infection. And just like how we can have immunity to a virus that we've been infected with, so we don't get reinfected, the same can be true for bacteria. So it can survive this and have an immunoprotective effect from future infections. But there's another effect from all of this, and that is called phage conversion. This bacterial chromosome, represented with the purple here, now has a new set of genes represented with the pink here. So its entire set of genes has been altered with this new introduction of DNA, and therefore it might have some different traits than it would have had otherwise. And that's called phage conversion. Phage again means virus, so it's been converted by that virus. And then the third thing is called specialized transduction. Transduction is the introduction of more genetic information to a bacteria from a virus, and it can also be called induction. And this virus, sometimes it doesn't just take its own viral genome, shown in pink here, but when it gets induced and it gets clipped out of the bacterial host genome, sometimes it makes a whoopsie and it grabs some of the bacterial genome as well. So now it's a vector. It's carrying around that bacterial gene, and it injects its new bacterial host. And guess what? When a lysogenic phage cycle occurs here, not only did it bring in its own viral DNA, but it introduced new genes from a different bacteria. And so this is a way that bacteria can obtain different genes that might have better functions or allow that bacteria to perform new tasks. And so this can advance the evolution of that particular bacterial organism or a population of bacteria. And that is called specialized transduction. When we learned about mitochondria, I said, it's good that it's kind of interesting to take a step back and think about who we are when each one of our cells has a mitochondria inside of it, or one or more mitochondria, and those mitochondria are the descendants of bacteria. So we have these bacteria, in a sense, that have evolved to live inside of our cells, and that's kind of an interesting sort of introspection that we can have by looking at these things. And I want to do it again here. We just learned about the lysogenic phase, and there are these viruses that can infect animals. They're called retroviruses. We'll talk more about those later, as I said we would. But within our DNA, there are these elements of DNA called transposable elements. And these are called jumping genes. And they have this interesting property that they can copy themselves in the different places in our genome. And it's really bizarre that that is something that occurs because that could really wreak havoc. If it goes inside of one of your essential genes, it could potentially be catastrophic. But these transposable elements, they seem to have obtained that trait from maybe what they were originally designed to be. And they're actually related to retroviruses, which again copy themselves into their host's genome. And what's so incredible about these is that up to 45% of our DNA is composed of transposable elements. So not only do we have mitochondria in each of our cells, but we also have about half of our DNA are made up of transposable elements. So let's switch gears here, and now let's talk about animal viruses. Naturally, their hosts are going to be animal cells, and the type of animal cell, their host range is going to vary depending on each virus. They, unlike the bacteriophages, they have much more variety of morphologies, and there's multiple types. We have the viruses that can have DNA genomes, RNA genomes, and then a special type that kind of oscillate between the two called the retroviruses. Animal viruses follow the same basic steps as a bacteriophage in their replicative life cycles, but there are some variations owing to the difference in anatomy of the host and the virus. So for attachment, we saw bacteriophages use their tail fibers to do that initial interaction with receptors on the bacterial cell wall. Animal viruses, on the other hand, are going to use spikes or fibers coming off of their membranes to interact with the receptors of specific receptors to their host cells' own membranes. For penetration, we saw that bacteriophages actually they contract and inject their DNA into the host cell. With animal viruses, the entire capsid will enter the cell either through a process called endocytosis or fusion. During the penetration step, the DNA is injected directly into the cell, and whatever's remaining of that bacteriophage just falls off. So there's no uncoating process of the bacteriophage's membrane or anything like that, or from the capsid. But with animal viruses, as we said, the entire capsid enters via endocytosis or fusion, and that can include coming in with a membrane. And so now we've got to shed off all of that material so the genome can get to work. So the genome has to get released from the capsid. In bacteriophages, the DNA is transcribed and translated. You might have a lysogenic phase, but again, during biosynthesis, you're going to have that transcription and translation that occurs there. And with animal viruses, it's going to vary a little bit depending on the virus type. So if we start with DNA, then okay, it's going to be very similar to bacteriophages. We're going to transcribe and translate. But if you have RNA already present, then you're not really transcribing. Transcribing is when you make an mRNA copy of DNA, right? And retroviruses is a whole different world. We'll talk about that one after we talk about DNA and RNA viruses. And then we have maturation. Maturation for bacteriophages is when we have this auto-assimilation of the different the bacteriophage parts. But with animal viruses, it can be a little bit more complicated, and it might require the use of enzymes in order to catalyze those different reactions. Release basically always kills the host cell for bacteria. Um, this may not always be the case for animal viruses. They might not always lead to complete lysis of the cell. So let's talk about fusion and endocytosis, the two ways that these animal cells gain entrance into the organism. So here at the very top, we see that we have our enveloped virus. And our enveloped virus, it has a membrane, a lipid membrane that surrounds that capsid. And when that lipid membrane comes in contact with a host cell, that lipid membrane, which is very similar to the host cell's lipid membrane, it's going to auto assimilate with those phospholipids, just like a phospholipid layer auto assimilates. So you can see that this bottom lipid that comes in contact with a host cell, it's going to blend in with a host cell. Like if you imagined if you blew bubbles and one bubble landed on top of another bubble, they could fuse together. That's exactly what's happening here. It fuses together and it makes this pore that the capsid then can be released into the host cell. Now, for non-enveloped viruses, it's a little bit different. So we still have some spikes or fibers that allow it to identify its host, binds to those host receptors. Um, but there's no fusion that takes place. And so instead, it does this endocytosis. So the plasma membrane gets pushed downward into the cytosol, and then it kind of pinches off on the inside and can get released inside of the cell. This can also occur with an enveloped virus as well, but non-enveloped viruses can only go through the route of endocytosis. Now, as we've discussed, viruses can only replicate and do metabolism inside of a cell. And so we would, just like we would say that there are obligate aerobes or obligate anaerobes that can only live in the presence of oxygen or the absence of oxygen, they would be considered obligate intracellular parasites. They can only be grown inside of other cells. And when we want to study viruses, therefore, we have to grow them inside of cells. And in this image here to the right, you can see that there's an egg cell which can be infected with virus particles in order to be studied. And this can be important also for vaccine development, where you have to grow up the virus particles. So eggs, chicken eggs in particular, are a very common method for cultivating these viral particles. In particular, for the flu vaccine, is going to be produced via this method. All right, we've talked a little bit about these protein spikes, these fibers that come off of the viruses that help them identify their host range. And now we're going to learn about the two of them that are in animal viruses, the H spikes and the N spikes. The H spikes, it stands for hemagglutinin. So hemagglutinin is this protein spike that allows the virus to identify the host cell. And then we have the N spikes, and this stands for neuraminidase. Neuraminidase is this enzyme. You see it ends with ASE, so that's how you could remember that this one is the enzyme portion. And this allows that virus to break down the host membrane in order to be released inside of the cell. And these H and N spikes are what defines the host range of influenza viruses, for example. So you will often hear in the news the reference to a particular strain based upon its H number and its N number. So scientists number these according to when they've been discovered, and those particular H and N spikes allow it to infect different organisms. Now, when we develop a vaccine for the flu virus called the influenza virus, that will only work for a short period of time. As that flu virus multiplies within the cell, there are going to be mutations that occur. And viruses are known for being fast replicators and fast mutators. And for this fact, they're going to evolve quite rapidly. As they change in their viral particles, in particular the ones that are around the outside of the viral particles, so for example, those H and N spikes, the immune system can no longer recognize them based upon looking at their surfaces. It's almost as if they put on a new mask and we don't recognize them anymore with that new mask on. And so a new vaccine has to be developed every year. So about a year in advance, scientists do their best to predict which strains or clades, these are just substrains of flu virus, are going to dominate in the upcoming season. And they historically do this by looking at which strain has been dominant in the southern hemisphere if they're developing vaccinations for the northern hemisphere, because in the southern hemisphere it'll be flu season before it's going to be flu season in the northern hemisphere. And they try and make the best prediction there. Um, Particularly tricky strains to develop flu virus vaccines for is the H3N2 one. This is particularly difficult because it mutates in ways that makes it really hard to make a good match in the vaccine. Um, the H3N2 strain, for whatever reason, it tends to make mutations. Mutations are more common in chicken eggs. And so when we grow these in chicken eggs, we tend to have a big issue with just getting the wrong mask in order to train our immune system against. And we are slowly improving in our methods. So we've started working actually with canine kidney cells instead of chicken eggs as a way to grow up these viral particles. They tend to mutate a little less rapidly in these canine kidney cells. So we're getting a little bit better in our methods. We also have some antiviral drugs that work on viruses. And the way these viral drugs are discovered is we're really trying to go after those different parts in the viral replication cycle. For example, Tamiflu inhibits the release of virions by interfering with that neuraminidase, the N spikes. Again, the neuraminidase helps to dissolve the membrane to allow for release of the flu virions. And here we have a medication called zidovudine, which inhibits the integration of HIV proviruses by interfering with reverse transcriptase, which we'll talk about in a little bit here. Another thing we can do is create nucleoside analogs. So we have these two different components here. We have this medication called remdesivir, and this is a ribonucleotide analog. And what it does is it imitates adenine nucleoside triphosphate. And this particular nucleoside analog, it interferes with viral RNA polymerase function by mimicking this particular nucleotide adenine. See how it's very similar to the adenosine triphosphate shown on the right side. So far, we've just discussed viruses, but as I said, we're going to cover viroids, virusoids, and prions. So let's go ahead and just cover those briefly here in the next following slides. So viroids are one of the smallest viral particles out there. They're just a short strand of circular RNA. So unlike viruses, there's no capsid there. And they can't, just like viruses, they can't replicate without being inside of a host cell. And these are usually plants, as is true for virusoids. And so they're responsible for causing a lot of plant diseases. So they're still very important to study. Now, virusoids, they're just a single-stranded piece of RNA. And again, unlike viruses, there's no capsid. Virusoids are even simpler. In fact, they lack some of the necessary components in order to replicate. They actually require a helper virus. So they must piggyback along with another virus in order to be an infectious agent. They have no nucleic acids. So this is a really bizarre infectious agent. How can you replicate and go on to infect more organisms and reproduce if you don't even have the genes in order to do so? Well, it's a little bit strange, but these prions are abnormal folding proteins that are going to be present normally within that organism. So these proteins, they're typically involved in the brain, and if a prion is present, it's going to cause some sort of neurodegenerative disease. So here we have on the right-hand side a basic mechanism of how this works. Here we have a normal protein in the brain of an animal, and that normal protein, there is another way that it could be folded. They can be a misfolded shape, shown here on the right side. This would be the disease-causing prion shape. So most animals out there are just going to have the normal protein shape. But if it gets misfolded for some reason, that can be a problem. Now, lots of proteins get misfolded as they get used. Sometimes they get worn out and they're not folded correctly anymore. Sure, but these particular misfolded proteins that are prion disease-causing proteins, they actually cause a chain reaction. So if you look below, here is the correctly folded protein and a misfolded protein. And if this correctly folded protein gets in contact with a misfolded one, it causes that correctly folded one to become misshapen and shaped like the prion disease causing one. And those misshapen proteins can go on to find other healthy proteins and cause them to be misfolded, and so on and so forth. So that misfolded version spreads throughout the cell. And this can cause issues with the normal function of that cell. There's a lot of diseases, prion diseases that are out there. There's a mad cow disease caused by scrappy. There's Kuru. There's Creutzfeldt-Jakob disease. There's fatal familial insomnia. There's one that has evaded me just now that is infecting deer and is a big concern by wildlife biologists currently. And another thing that's interesting about these prions is these disease-causing prions are very heat resistant. So you can't kill them just through boiling or normal cooking methods, and they can survive for a long time in nature, which makes them hard to prevent their to disinfect from prions. So another thing about these prions too is they can be both transmissible or inheritable. So you can imagine that if a host is infected with these prions, that they could potentially pass it down to their offspring. Kuru has an interesting history. It was discovered in a tribe in Papua New Guinea. And this disease was actually linked to cannibalism of brain matter in the 50s. So when people would eat the brains of other people, they would get those prions, those infectious prions, and then they would become infected. And it was pretty customary for women and children to do this ritual. They thought that it was a way to release the spirit of dead members of the tribe. And so it was a big issue, but through education, we were able to correct this and reduce the presence of Kuru in these populations. All right. So let's talk about I said that depending on the type of nucleic acid they have, these viruses are going to have different methods of their life cycle. Although they're all going to be the same basic steps, It's the way they do their transcription and translation really is going to vary between the two of them. So single or double-stranded DNA viruses will transcribe their DNA to RNA, right? Their genes into RNA or mRNA, and then those mRNA um, um, signals can be sent to the ribosome to make the genes necessary. But viral RNA is translated in the cytoplasm, and all viral components are assembled to maturity in the nucleus. Whereas that's not the case with viral DNA. You are going to have to convert that, and you're going to have to do DNA replication there. So let's go through this viral DNA. As it enters the nucleus, we can see there's that small purple ribbon. And then the early genes for hijacking the cell, they're going to be transcribed into mRNA, and then they're going to be translated by the ribosomes. And then we'll have some later genes that are then going to be transcribed and translated that are not necessarily for that initial hijacking, but for making more viral particles, for making the enzymes required for assembly, so on and so forth. And then the virions are going to be assembled and released. That's pretty similar to what we saw with bacteriophages. The most important DNA families that you need to be aware of are going to be the Adenoviridae, and these are going to be responsible for causing acute respiratory diseases such as the common cold. Next is the Poxviridae, and this can cause diseases that are associated with skin lesions, such as smallpox and cowpox. Next is Herpes viridae, and these are associated with causing sores or rashes, including cold sores, chickenpox, mononucleosis, and roseola. Next is Papoviridae, and these are associated with causing warts and tumors and may result in certain types of cancers. And finally, we have Hepatnaviridae, which causes hepatitis B, which is actually unrelated to other hepatitis forms, but Hepatnaviridae causes hepatitis B, which is a big issue in this country and around the world. Viruses can, because they are so good at transmitting nucleic acids from one organism to another, they can actually be used for gene therapy. In particular, the adenoviruses, several of them are harmless to humans, but they do infect primates of other varieties. And so we can take these adenoviruses that are capable of propagating genetic information in human cells, but aren't really disease causing, and we can put in particular genetic information that we want to treat disease. So in this example here, we have the first gene therapy that was created to treat an eye disease, and its method of delivery in this vial here are the virion particles of adenoviruses with a gene correct gene flaw present in certain individuals. All right, so now let's get into the RNA viruses and their genome structure. So RNA viruses, their genomes can be double-stranded RNA. And they could also be single-stranded. And when they're single-stranded, they can be made up of the template strand, or they can be made up of the coding strand. So we would say that they could be single-stranded a positive sense or single-stranded a negative sense. The positive sense acts as if it was just mRNA. It can go to the ribosome and be translated into proteins. The negative strand cannot. It doesn't make sense it's a negative for making sense to the ribosome. It has to be it needs to have a copy made of it, of the other complementary strand, and then sent to the ribosome. It's probably a little confusing here, so let's go back to our strand naming conventions. Now, with our strand naming conventions, when you're looking at DNA, we describe the template strand is the strand that's used for doing the transcribing of DNA to mRNA, right? That's the template for making mRNA. And it's anti-parallel and complementary to the coding strand. And so the coding strand would look just like the mRNA strand, except for instead of having U's, it would have T's. And the template strand is the strand that marries up to it. So let's look at instead, and you can go back and review that with microbial genetics chapter if you are a little bit fuzzy on that before moving on. It might help you make sense of all of this. But let's look at that strand of RNA from a virus. So let's say that virus has a negative sense, sometimes called an antisense or negative sense strand, single-stranded virus. So here's its genetic code shown along the bottom. And when it's a negative strand, this RNA is in the wrong. It's a wrong strand to be sent to the ribosome. If we were to send this strand to the ribosome, the ribosome's not going to be able to make any proteins out of it. In order for the ribosome to make proteins from this genetic information, we first need to make a copy of it. Okay? So just like with our DNA template strand, the negative sense strand has to have a copy made of it. Okay? And now that copy could be a messenger RNA that can go to the ribosome. So I always remember it is if this is a negative sense, when it gets sent to the ribosome, it doesn't make sense to the ribosome. Positive sense strands make sense to the ribosome. And so if you have a negative sense stranded, single-stranded RNA virus, you have to make a copy if you plan to send it to the ribosome. You have to make that coding strand version of it. Now, some viruses, they are ready to hit the ground running. So they already come as a positive sense strand. And so this is synonymous with being an mRNA strand. They are ready. See this AUG, that's the start codon. It's ready to go right to the ribosome. The ribosome is going to identify that start codon and start using the tRNA in order to build out that protein strand according to this template strand. It's ready to go. All right. Now, this is going to cause also challenges for replicating the viral RNA. See, in the complete virion, all of the RNA in that complete virion, if it's single-stranded, all of it has to be antisense, if it's an antisense or a negative sense virus. Or if it's a positive sense virus, all of its strands of DNA in that virion particle are going to be positive sense. And so if you need to make more virion particles from this original strand here, you have to copy it into this new strand, the template strand, and then from here, you can make the final RNA strands. And hopefully we can see what that looks like here on rules for RNA virus replication. So every new virus that's going to be made in that cell, it's going to need some RNA and it's going to need some protein, right? It's going to need some protein to make its capsids, some enzymes maybe to help with assembly, and it's going to need its own genome, its own set of RNA. So if you have let's say you have a positive strand RNA. Remember, this is synonymous with a strand of RNA that when it's injected into its host cell, it can go right to the ribosome and start making the proteins necessary. It doesn't have to make a copy of itself first. In order to make the final RNA for new viruses, well, it has to first make a negative strand, and that negative strand can then be a template for making more positive strands again. So it takes its original RNA strand, which is a positive sense strand, it makes a negative sense strand, and that negative sense strand is then used to make the final positive sense strands. Because remember, replication is semi-conservative. It always makes the opposite strand. So it can't use its original strand to make an original strand. It has to make the other version to make the original version. It's kind of weird, I know. But remember, it's always going to be making the opposite strand. That's how it copies. So it makes that opposite strand so that it can make the original strand. Again, just to reemphasize this point, only the positive strand can be used to make protein. So this original strand that was infected into the cell, that can be used to make protein. All right, so let me show this again in a graphical depiction. So here we have our single-stranded positive sense RNA virus nucleic acid. It's freshly into the host cell, and it can be used immediately to make proteins, right? Great. We can make the viral proteins, excellent, like the capsids shown here, and your spikes, and your H spike and your N spike. But it cannot be used to make more positive sense strands, not yet anyway. It can be copied into a negative sense strand, and that negative sense strand then can be used to make the positive strands, which are then used to be packaged into new viruses. Now let's look at the single-stranded negative sense RNA viruses. They inject their negative sense RNA into the host cell, and they cannot go straight into making the capsids or the N or the H spikes or any enzymes, because this, in its current form, does not make any sense to the ribosome. It's negative for that. So first, it makes a copy of the RNA into a positive sense strand, which can be then used by the host cell's ribosomes to make the proteins, and then can be used also to make the negative sense RNA strand. And these, we combine them together and we can make the viral particles. So at the end of the day here, you should be able to say, okay, if I give you a positive strand RNA virus or a negative strand RNA virus, you should be able to tell me the order. If you have a positive strand, can you make proteins right away? Okay, yes or no? Yes for a positive stranded RNA, no for a negative sense stranded RNA. And then what steps are required to generate the final nucleic acids that can be packaged into virion particles? Well, both of them have to copy into the other strand and then back into the original strand. Okay, and then finally we do have the double-stranded RNA viruses. This one is a little bit less complicated. Much like we saw with DNA replication, you're going to need to make a semi-conservative replication of both of those individual strands so that you have double-stranded DNA. So once you make the copy of each one, it's not shown very well here, but you got to make a copy of each side, and then that double strand can therefore be packaged away. Okay, let's now get away from looking at single-stranded RNA viruses and double-stranded RNA viruses, and let's go look at retroviruses. Retroviruses, they have RNA genomes, but they also have a special enzyme called reverse transcriptase. All enzymes that we've looked at so far, they either convert DNA into DNA, as we saw with DNA replication, DNA into RNA, which we saw with transcription, or RNA into RNA, that's RNA replication. But retroviruses can do something incredible. They have this other polymerase called reverse transcriptase. It does reverse transcription. So instead of converting DNA to RNA, it converts RNA back into DNA. So the viral DNA can then be integrated into the host genome. So we have typical viral penetration into the host cell. But once in there and we've uncoded the capsid, we've released the viral RNA, the reverse transcriptase enzyme converts that RNA into DNA, and then that DNA gets integrated into the host cell genome. We call that protein, that enzyme that does it, we call it integrase, simply enough. And now we have a provirus. And that provirus can then be transcribed and translated into making viral proteins. And then another enzyme comes in called protease, which helps to assemble those viral components. And then that virus can be released from the organism. It can be released through something called budding, which we talked about with the viruses some. It's a little bit different, but it basically is a outgrowth of the original plasma membrane, or it can be released through lysis. This is basically just an example of how that budding works. So here we have a viral capsid, and it's added viral proteins into the plasma membrane of its host cell, and it is able to have this outgrowth against the cell's plasma membrane, and then it pinches off here at the back end, and that plasmid creates its own lipid membrane around an envelope around the viral capsid. And you might be wondering, why is it so important to be familiar with all of these different steps of these different organisms? Well, when we want to create drugs in order to treat these viruses, like we showed with attacking the H spikes or the N spikes of flu viruses, our drugs are developed in order to attack these different steps in the virus life cycle, quote unquote. So, for example, we do have antiretroviral drugs that attack the reverse transcriptase enzyme. We have drugs that attack the integrase enzyme that allows it to integrate into the host's genome. And we also have some that might attack the protease enzyme that allow for the assembly of the viral components. So if you don't know how the viral life cycle works, you don't know how to apply medicine in order to fight and combat these viruses. All right, now I said that we are going to cover acellular pathogens, and then we were going to loop back and finish a little bit more about microbial genetics. And that little bit more that I wanted to discuss was gene transfer. So, prokaryotes are just like eukaryotes. They need to make sure that they're acquiring genetic diversity so that they can evolve and adapt to new environmental conditions. And genetic diversity can occur in a couple of different ways. One way is just the cellular machinery that copies our DNA during DNA replication can make mistakes. And that those mistakes can be good or bad, and they can be passed off to the offspring. And it's a bit of a crapshoot. You may have got a good one or a bad one. And that happens at a common rate of roughly 1 to 3 nucleic acids per generation. So in us, each one of us is a mutant, so to speak, where there are going to be 1 to 3 mistakes in the genetic code made by your parents when they produced the gametes that led to you, and they're going to recombine, to form your initial cell and zygote. Another way that we get genetic diversity is through sexual reproduction. That's the meiosis portion of our reproductive cycle. Well, how do prokaryotes get their genetic diversity? Yes, their cellular machinery makes mistakes when making copies of their DNA, but that's not enough, right? We do have those mutations that occur, but how do they acquire completely new genes like we do, right? We develop sexual reproduction. Well, Prokaryotes have horizontal gene transfer. Horizontal gene transfer. And in fact, our sexual reproduction, it's hypothesized that we developed it because when we transitioned to having organelles that housed our nucleus, we could no longer do horizontal gene transfer like bacteria can. And sexual reproduction evolved as a way to encourage genetic diversity. Well, we don't know what horizontal gene transfer is just yet. It's kind of a strange way to describe it. And maybe a great way to learn about horizontal gene transfer is to see what it is not, and that is vertical gene transfer. Vertical gene transfer is the passage of genes from one organism to its offspring. Why is it called vertical gene transfer? Well, if you imagine if you were to draw your family tree, you would draw it vertically, right? You would start with the oldest generation at the top and then the next generation below that, and you draw lines connecting them, so on and so forth. So those genes are transferred vertically from parent to offspring. And with horizontal gene transfer, it does not require a new generation. This is where two organisms get together and they can share genetic information between them. There are three modes of horizontal gene transfer that you need to know. It's called conjugation, which is just as it sounds, transformation, and transduction. So conjugation is, if it helps you remember, think of a conjugal visit. This is where bacteria get together and they transfer plasmids through direct contact. They transfer this information through pilus. It's called a sex pilus. We learned about pili in chapter 2 or chapter 3, maybe. You should have covered it in Bio 181 as well. And these pili make contact from one gram-negative bacteria to another, and through that pili is a hollow core, and a plasmid can be transferred through that hollow core from one of the bacterium to the other. Gram-positive bacterium do not have these sex pili. So gram-positive bacterium can still do conjugation, but rather what happens with gram-positive bacterium is they come together and their cell surfaces stick to each other, and that forms what's called a mating bridge. So they come in much more closer contact with each other, and that allows that conjugation to occur. This requires a certain genetic chassis in order to build out that sex pili and allow the transfer to occur over. It requires a fertility plasmid. We abbreviate that to F plasmid. You can see highlighted in the red box here. If a bacterial cell is F positive, meaning it has that plasmid, it's capable of forming a sex pili. And so therefore it can donate its plasmid. So we can see here's the F plasmid. So it has the genes required to grow that sex pili. And when close enough to another gram-negative organism, it'll build out that sex pili and create contact with that recipient cell, who is going to be F negative. And that creates what we call as a conjugation bridge. Now, the bacteria doesn't want to lose its F plasmid, so it just makes a copy of one of the strands and sends it across. Okay, it doesn't go through the full semi-conservative replication and copy both strands. It just copies one strand and sends it on over, and we call this the rolling circle form of replication. Some viruses use this as well. It's something you should be familiar with. You can think of it as it kind of looks like a toilet paper roll. Sends over that newly synthesized DNA strand, and then the second strand synthesis can begin by the other organism. And now you have both F positive organisms in both cells. And it can also send over other genes, some common genes that it may send over are different virulence factor genes that allow the cells then to be more infectious or more virulent, we would say. So genes for varotoxin, which causes dysentery, can be sent from S. dysenteriae over to E. coli. Or the genes for tetanospasmin toxin or tetanus can be derived from certain C. tetani. This is probably the one that concerns most of us the most, and that's antibiotic resistance genes. So during that rolling circle replication, a gene can be sent over that might allow that organism to be resistant to methicillin. And that's where we get our Staphylococcus aureus, a common microbe that grows on our skin that might be MRSA, resistant to methicillin. And also one of our other important antibiotics, vancomycin. There are vancomycin resistance genes present on plasmids that can be shared through this horizontal gene transfer, leading to MRSA Staph aureus. So conjugation is one method. Another method is called transformation. And during transformation, this does not require a donor cell and another cell in order for this to take place. This is just taking up what we call naked DNA. So in the environment, there are all sorts of different, remnants of cells out and about, and sometimes there's just pieces of DNA. And those pieces of DNA can be swallowed up by a recipient cell. And sometimes that DNA can be genetically recombined by that recipient cell into the host chromosome. And when it does so, we would call that a transformed cell. Its genetic material has been transformed. And through this process, it can gain new traits, and we therefore call that a transformation process. And some bacteria, some microbes, some cells are more resistant to this process taking place. They don't want their genome to be messed with, and some of them are more amenable and encourage it to occur. And finally, we have transduction. We've talked about transduction already. Transduction is when you use a virus in order to act as a vector to transport that genetic information. This can be done through a bacteriophage with bacteria or other viral animal phages as well, where that genetic information is brought in, it integrates into the host cell and host genome. And not all the times is that host genome destroyed in that process. It might survive and has that new piece of information inside of it. And this process is what we saw in that first treatment for the genetic disease, eye disease, where we used the adenoviruses in order to transfer that genetic information. And that wraps up our coverage for gene transfer from microbial genetics. Thank you so much for watching. I hope you enjoyed. Let me know if you have any questions.